DNA replication in prokaryotes involves a circular DNA template strand that produces a single replicon. The existing DNA and multiple enzymes facilitate the process of replication. The first stage of DNA replication is initiation. An initiator protein binds to the origin of replication, which causes local unwinding to make a short stretch of single-stranded DNA available. Prokaryotes only have one origin of replication in the DNA, and it plays an important role as the site of the start of replication. To further unwind the DNA, an enzyme called helicase, represented by the dark spiral pasta, attaches at the replication fork and breaks the hydrogen bond, holding together the two strands of DNA. Single-stranded binding proteins also coat the single-stranded DNA to protect it, stabilize it, and prevent it from becoming double-stranded. DNA gyrase, represented by the folded pasta, is another enzyme that plays an important role in DNA replication because it relieves supercoiling ahead of the replication fork. The next stage of replication is elongation. The template strand provides an anti-parallel sequence of DNA so that DNA polymerases can add the correct nucleotides in the 5' to 3' direction. To elongate the DNA, polymerases bond to the 3' hydroxyl group of the last nucleotide on the new strand to the 5' phosphate group of an incoming deoxyribonucleoside triphosphate. The two extra phosphates of the DNTP are cleaved off and a phosphodiester bond forms between the two nucleotides to hold together the DNA. The DNA polymerases that facilitate this process must have a free 3' end that they can add nucleotides to. In order to get the 3' end, RNA primers are made by primase, a type of RNA polymerase. Primase, represented by the cut spaghetti, plays an essential role in DNA replication because it provides a 3' end for nucleotides to be bonded to by DNA polymerase. DNA polymerase III in particular, represented by the shell pasta, adds nucleotides to the primer in the 5' to 3' direction and then proofreads with exonuclease activity in the 3' to 5' direction. DNA polymerase I, represented by the light spiral pasta, functions in removing the RNA primer and also proofreading the nucleotides in the 3' to 5' direction. If DNA polymerase I could no longer perform its exonuclease function, it would not be able to proofread nucleotides in the 3' to 5' direction, which would most likely lead to errors in the DNA. DNA polymerase I would also not be able to remove RNA primers in the 5' to 3' direction, which would prevent the DNA from being fully replicated since the RNA gap would not be filled with DNA. The leading strand during elongation is the strand of nucleotides that is continuously moving towards the replication fork. The strand in the 3' to 5' direction is the template for the leading strand since the new strand will be in the 5' to 3' direction, with nucleotides being added to the free 3' end toward the replication fork. In contrast, the lagging strand is the strand that is being made in fragments since the 5' end is toward the replication fork. These fragments are called Okazaki fragments and must be formed since nucleotides can only be added in the 5' to 3' direction. After several Okazaki fragments have been created, they are sealed together by an enzyme called ligase, represented by the cut fettuccine, after DNA polymerase I has removed the RNA primers. Since the DNA that is replicated in prokaryotes is circular, replication ends or even repeats when all of the nucleotides in the circle have been added to the template strand.